Yeah, I guess we can start. It's 12:15, right? Okay. All right. Thank you, everyone, for coming to this last biology club for this year. And first of all, really sorry about the, I don't know, 10 or 12 or 15 emails that you received. And because the email lists were not working, and apparently they just fixed it. And so all of them came together. And so today we have a talk from Nicolas Cuny. He's a postdoc in our lab, so in the lab of Guillaume Salbreux. He did his PhD from Grenoble, and he did it in biological physics. And here he has been working since last year on these active surface models and how to apply them to biological tissues. And so, yeah, you can hear more about it.
Okay, so hello everyone. I'm pleased to have this opportunity to present you part of my work in active surface models. So as Ria said, I'm a postdoc for now 1 year in the group of Guillaume Salbreux. So we are located at Sciences 3 in the third floor. We have 2 PhD students, Simone Ciccolini and Mukund Kotari, and the 4 postdocs, Quentin Wein, Ria Nandé, Jérémy Frankfurt, and myself. And we are all physicists, so we are not doing pure biology, but we try to apply some tools of physics to biology. And one purpose of my talk today will be to show you what kind of knowledge, what kind of information we can get from physics on biological systems. And I will focus mainly on active surface models that are a specific field of biological physics. So what are active surface models? So surface models is not something that is specific to biological systems. It's been developed for more than 1 or 2 centuries now. And it's a way of describing the mechanics of all kinds of materials. So the idea is the following: as you may know, for every material you can find a scale where it is discrete, and you have a lot of constituents, for example, atoms. And if you are doing mechanics at this scale, you have to deal with all these constituents, all their interaction, how they are interacting all together. So this is a huge number of information that you have to get. But hopefully, as you see at our scale, all the matter seems to be continuous. Okay, so the idea is the following: is to go at a high enough scale in a way to have some continuous description of the medium. And now you are just forgetting all about the individual components, all about the interaction, and you are just describing your material as a bulk with some mathematical tools. That is the stress tensor, that is mainly giving you the internal forces at some point of your material. So to characterize the mechanics of one material, you have to get some law to say how it's behaving and how it's responding to deformation. So you have to empirically determine some relation between the deformation, so that is enclosed in this deformation tensor epsilon, or the rate of deformation, that is just the derivative of it, the speed at which, the velocity at which the material deform. And you have to get some relation and to say how a deformation on the rate of deformation is generating internal force. So this is a relation between those different elements, but to give you the stress tensor as a function of the deformation of the rate of deformation. When you have a relation between stress and deformation, we call that elasticity. When you have a relation between stress and the rate of deformation, we call that viscosity. And in general, in most of the case, all materials have both elasticity and viscosity. Once you have that, you can just write an equation on the stress tensor that gives you kind of equivalent of the second law of Newton, the equilibrium of forces. All the forces has to be balanced. And when you write that, as you are using the relation relating stress to deformation, you get some equation on the deformation, and you get the shape of your material for a given configuration. But you can go even further, because in some material, you have one dimension that is thinner than all the other. You can think of a sheet of paper, for example, it's quite flat. So there is one direction where you don't care about what is happening. So if you have such a dimension, you can just forget about it and just describe your material as a surface, an infinitely thin surface. And now, if you want to describe the mechanics of such a surface, you have to work with two new tools, two new objects that are the tension tensor and the torque tensor. Now, I just would like to give you some idea of what are those tensors and how they relate to stress, and why do we need two tensors? Because for bulk, we just need one tensor. Now we need two. So what is the point behind that? Just think about a 3D material to which you are applying a stretch at the top, on the compression at the bottom. Just the same amount of stretch on the compression to be simple. Here in blue, you have the mean surface by which you would like to describe your material. So if you take some simple linear material, what you will get, if you are looking at the stress, at the internal force, you will just have some gradient of forces there. And you have to reduce this mechanics to just one surface. So the first thing that you will use to describe the mechanics is just to sum all the stress, and this will give you the tension tensor, okay? But as you see there, if you are just summing all the stress, what you get at the end is just a zero tensor, a zero force, because you are summing a vector that are opposites. So if you do just that, if you are just looking at the tension tensor, at the level of your surface, there is nothing happening. So what is important here is that you have some inhomogeneity of your stress all over the width, and that's what is catching this torque tensor. It's some object that is giving you an idea of how heterogeneous is your stress on the width, and you see that it will tilt the boundary of your material, so you will have some bending. So to be simple, you can just see those two objects like that. The tension tensor is something that telling you how the forces are stretching or compressing your surface in its plane. And the torque is telling you how the forces are bending your surface. So just like for stress tensor, you can just write some relation with the deformation and write the equilibrium, and this will give you the shape of your surface at all time in all configuration. So that was about all the surface model. And now, how can we use it for tissues? So we can just play the same game. If you have a cell tissues, if you are looking at the cell level, the tissue is just discrete, made of a lot of cells that are interacting all with other by a cell adhesion, for example. But once again, we can just go at a high enough scale and see that as a continuous media. And in most of the case, and in particular for monolayer tissue, you see that your layer is really thin in one direction and expanding in the other. So it's quite relevant to describe your tissue as an infinitely thin surface. So it just, as I presented you before, you have your tension tensor, your torque tensor. You are just writing some relation with the deformation. But now, what is different with tissue? We didn't take into account up to now the biology of the tissue, because tissue is not like a sheet of paper or this table. It's not an inert material. It's a living material, what we call an active material in physics. And in particular, it's able to consume some energy to self-generate forces inside of it. For example, you can just think of the cytoskeleton of cells that is able to consume some energy to create forces. So we have to take that into account. For now, we had some passive parts in the tension tensor and the torque tensor, but we are just describing how the tissue is responding to deformation. And we had just some terms that are active terms on the tension and torque tensor that are taking into account those kind of phenomena. So once again, you can write the equilibrium for force on torque, and it will give you the shape of your surface. So the way we describe this active phenomenon, we kind of separate it in two kind of effects. As you see, for example, if you look at this cortical actin or this kind of organization, all the cytoskeleton is going to pull on your cell on the same way, whatever the direction. It's what we call an isotropic effect. Whatever the direction you are looking at, the tension and the torque are the same. But in a biological system, most of the time you also have some anisotropy. You have some orientational effect, collective effect. For example, you can see there in some tissue you have a shape anisotropy, and all the cells. The cells tend to elongate on a direction, and they try to orientate the direction all together. So that can be a kind of anisotropy you see in a biological system. You can also think of anisotropy coming from the biology, just like a planar cell polarity, where you have a gradient in the concentration of some proteins that give you some preferred direction. And most of the time, those anisotropy also generates tension on the torques that are also anisotropic. So that's the second kind of effect that we can look at with a active surface model. It's anisotropic effects. In this case, you will have some preferred direction in which the cell is pulling. For example, on it will push in the orthogonal way. So that's about active surface models. And once you have that, you can try to understand how these active effects affect the shape of tissue, and to get an idea of how cells are generating the right forces to get the right shape, in example for morphogenesis. So now I will just show you some example of application of active surface coming from our own work. So we have some collaboration with people in Nice, Barthélemy Delorme, Grégory Mandelin, Jennifer Cochet, and Matteo Rausi, that are working on the sea urchin gastrulation. So sea urchin is an animal living in sea, and at the early stage of its embryogenesis, it's performing gastrulation. So after like a 10 cell division cycle, you get something that is quite spherical. You have some monolayer tissue that is quite spherical. And at some point, it starts to flatten at a pole, and even to inward folding, creating a tube inside the embryo that is going to reach the opposite pole, to stuck to the opposite pole, and that will become the primary guts of the animal. So here you can just see it on a video. So you have some cross-section of the sea urchin embryo. You have to think of it as axisymmetric. Okay, it's a revolution symmetry, and you see. So it's okay. It's frozen. It's really a monolayer. You see here the membrane. You see one cell in the tissue mainly, and you see it folding inwards at this point. Okay, so some vocabulary for what is next. When I'm talking of basal side, it means the inside of the embryo. Apical side is the outside. The vegetal pole is the pole at which the invagination gets developing, and the animal pole is the opposite one. Okay, so now we try to understand how the embryo, how the tissue is managing to get this form, and to inward folding to build this primary guts. So for that, we use some active surface model that has been developed by Diana Koromskaya. That was the previous postdoc with Guillaume Salbreux. And the idea is exactly the same as what I was saying you. So you have your embryo, you can just consider it as a axisymmetric tissue. And we just forget about the cells and all of that. We forgot about the bulk. We just describe it as an active surface. Okay, so here you have to think of this contour as a surface, an axisymmetric surface. And you can just describe it by, as I said, by the tension tensor and the torque tensor. The shape will be determined by your force balance, and now you have to choose what you put inside of these two tensors. So, as I said, there is some passive contribution, but is just describing the response of the tissue to deformation, and there is an active one for the contribution of the biology of the system. So, in the passive one, we mainly take into account 5 effects. There is a lot of effects that we can take into account, but we just focus on the main one and the more important one in a way to have something quite simple. So there is, as I said, elasticity. So when you are stretching or compressing the tissue, is going to generate some tension in a way to resist to this deformation. Just the same, you have bending elasticity. If you try to bend your tissue, it's going to generate some torque in a way to go back to its flat state. And you have also viscous effect that are coming with that. So I don't go too much into the detail of that. It's just a way of giving the rheology of the tissue, how it's responding to deformation. And what is more important is the active part. So just as I said, it can be either isotropic effects, all is the same in all directions, or anisotropic effects. And it can be either tension and torque. So in this model, you have 4 kinds of active effects, either tension or torque, or either isotropic and anisotropic. And as we put anisotropic effects, we have to say to give them a direction. We have to say how are they orientated. And for that, so for people that are used with this kind of terminology, we are using a nematic field that is minimizing some Frank, Landau, Deugen energy at the surface of our stuff. For those that don't know about that, we just take some orientation that is circumferential. So either the cells can pull in the circumferential direction, either they can pull in the radial one. It's because we have something that is axisymmetric, so it's not really relevant to have something that is orientated, breaking this symmetry. So, okay, that's all about the model. And now just see what we can do with that, what we can understand from gastrulation. So we are really focusing on this gastrulation part of the embryogenesis. And in particular, first, we would like to understand how the embryo managed to get from this spherical shape to this flattened shape. So for that, we go to the experimental observation of our collaborators. And when we are doing so myosin fluorescence imaging, what you see is that just before the beginning of the folding, the invagination, you have some excess of myosin there at the vegetal pole, and it is mainly apical. Okay, and if now we are looking at our embryo from the bottom, what we see, so it's still a myosin fluorescence image. So here you see the membrane of the cell. Here you have a cell, for example, and you see that you have your vegetal pole there, where the invagination is going to occur. And all around, you have there some really small cells. So they are apically constricted. So you see, this is a kind of a ring of apically constricted cells. And this, in our language, it means that you have some isotropic effects somewhere there. So because you don't have any orientation, all they are just shrinking in area. So it has to be isotropic. And one other point is that, as you see, and we can verify by doing a tension measurement by micropipetting, the tension at the apical side, so outside, is much more than the tension at the basal side. So just remember this explanation of the torque. When you have some difference in the stress profile inside of your tissue, you also have a torque that are generating. So it means that in our model, we have to take into account both tension on torque. So we just tried that. We take our spherical initial state. So once again, you have to remember that it's an axisymmetric surface. It's not just a contour. And we just apply some ring of active isotropic tension to that, and just simulating the equilibrium equation. We can have the shape at each point and see what is happening. And when you do that, you see that indeed you get some flattening of the embryo at the pole where you have your ring. And if we compare to experimental data, we are quite good with that. With the images, we are able to extract some surface, experimental surface there. So in particular, we are able to reproduce these two shoulders that we see in experimental data. Okay, so that's for the first step of the invagination. But now we are wondering what is driving the invagination, what is pushing inside the tissue. And so first, one scenario that we tested is maybe that it's just that the apical constriction is becoming bigger and bigger. When you look at the myosin intensity during the time, so here the invagination is starting, and you see that you have some peak of the myosin at the vegetal pole. So we just ask us if we are just increasing the effect, the magnitude of the isotropic effects, could we reproduce this invagination? So that's what we did. This is what we get when you double the magnitude of both isotropic and both tension, isotropic tension and isotropic torque. So you see that indeed the invagination is becoming deeper, but the shape is not so good. We are still increasing, okay? So there now you have those kind of shoulders that we didn't see in the experiment, but still the invagination is going on. But at some point, when you increase it too much, it's no more relevant with the experiment. You see that you are not deepening anymore your invagination, and you are even closing it. So mostly this mechanism can be the driving mechanism that is driving the invagination. So we have to look for some other effects. So if we go back to the observation of the sea urchin with the myosin microscopy, so you see you have your ring of apically constricted cell, and if you take care, in the cells that are outside this ring, you have some kind of orientation of the junction. You have some long cable, supracellular myosin cable that are forming. And if you look at those cables during the invagination, so you have here the invagination of the center. And in blue, we highlight one of those cables. Of course, there is a lot of other cables, but you can just focus on this one. And you see that during invagination, this cable is shrinking and converging to the center of the invagination, just like a string purse effect. So we suspect that this could be the driving effect for the invagination. And once again, we would like to test it with our model. So in this, how can we describe it with our model? It's something that is highly orientated. You have some anisotropic effect there. And you have tension, an apical tension that is greater than the basal one once again. So we should take that into account through anisotropic effects, both tension and torque. So what we did is just to take our previous shape, once you apply the isotropic tension and torque ring, and just apply some outer ring there with some anisotropic tension and torque effects. And when we are simulating that, what we see is that we reproduce the invagination, and you can have a tube growing up to the animal pole. So that is a good clue for this phenomenon to be the driving one. And now we just ask us what is the importance of this isotropic tension and torque ring? What if she wasn't there? It wasn't there. So we just do the same, starting from a spherical initial state, without applying first the inner ring, this purple ring there. We just put the anisotropic ring at the exact same place. And what we see now is that the tissue don't anymore manage to go inside. So this first ring is really important in a way to initiate the invagination. It has to first flatten the tissue, create some negative curvature, and after that, the outer ring is able to push on that and make the tissue going inside. You see, we reproduce quite well the experimental shape at some point on the invagination. And one other important point is that the inner ring, this ring of active tension on torque, is controlling the shape of your invagination. It's always at the tips of the invagination, so it's kind of fixing the radius of your invagination. So it also has an importance on the shape of your invagination. Okay, I'm frozen. Okay, so there is some nice experiment that highlight this importance of this inner ring of isotropic torque on the tension. If you take your embryo just sometime before the beginning of the invagination, so where it is flattened, but you don't have this anisotropic ring, you just have the isotropic ring, and you introduce some micropipette inside of the embryo. Now, if you are creating a deep pressure, what you see is that the embryo is once again invaginating. So it's quite important because there you don't have this outside ring, this anisotropic ring. So you don't have the driving mechanism, but just creating some other driving mechanism, you can obtain the same shape and the same control of the shape due to the inner ring of isotropic tension and torque. And that's something that we also see in our model. If we take the flattened shape and apply some negative pressure, you see that once again you have some invagination. So it's not so nice that the previous one, but once again you see that the inner ring is controlling the shape of the invagination. So it's quite important in a biological way, because in biology you always have some noise. So maybe the outer ring won't do exactly apply exactly the good strength, but thanks to this control of the inner shape of the inner ring, you will have a shape that is quite robust from one embryo to the other. Okay, so as a conclusion, I just summarize what we show with this model. So how does the sea urchin embryo manage to gastrulate? So first, starting from a spherical state, you have some ring of apically constricted cells, so cells that are generating isotropic tension and torque that will manage to flatten your embryo, create some negative curvature there. And now you can have some outer ring of anisotropic tension and torque that is going to push on this ring and make the tissue go inside. So once again, the important message here is that the inner ring is crucial to initiate invagination and to control the shape of the invagination, and that the outer ring is the driving mechanism for the invagination. So with that, I will conclude. Just some other active surface project that we have. Okay, I just show you an example, but there is a lot of work that we are doing in that. So we have here some tools. It's more a tool for experimentalists in a way to get some information on the mechanics of their tissue. So here you see the sea urchin embryo. We have some bachelor student that is working on coding this method. It's a method that is coming from active surface framework that we develop on that allows us to get some really precise mechanical information about the tissue. So if you have some tissue and you are tracking your cell in time, if you are interested in knowing about the mechanics of that, we can just talk, and we have some tools there to really quantify what you get. We are also working with Claire Dessal, that is now in the Roux lab, but working on with her on its PhD project that was about a blood vessel on ship. So we work with her experimental results in a way to try to build some blood vessel model to understand how the blood vessel responds to pressure. And one last stuff that we are doing for now is using the same model that I presented you for the sea urchin, but on other species that are nidarians, and in a way to understand the shape diversity all along this species in their development. So with that, I finish. I thank you for your attention. And if you have any question, I can try to answer it. Yes.
When you look at the animal shape before the being of the there is a change in tissue thickness when you have on the vegetal side. The height of the tissue is also a bit present on the animal wall. This change in height, you can imagine affects the torque.
Yes.
Would there be a way to, for example, to inhibit this change of height and see before the torque is still the same and causes the deformation?
I'm not a biologist, so I don't know if there is a way to do that, but I expect it's quite hard to do that, maybe with a good drug, but I'm not a specialist of that. But still, okay, so it's true that the thickness will have some importance on the torque, but what is really important for the torque is just to have some difference between the apical side and the basal one. And this is not changing with the thickness. It's just the value of that is going to change. So, for example, if here you don't have any torque, if you get thicker, you won't have any torque anymore. So it's just affecting the amplitude of your torque, but not the position of it on the surface. So I don't think that it will change too much thing, but it's also something that people have to deal with when you are doing the surface, because as I said, we are just forgetting one direction, but sometimes there is a really important thing happening in this direction, so you have to get it in mind when you do it. Yes.
A question on the first part, just flattened. You matched pretty well the bottom flat part, but you see that in the simulation, you also have the top is a bit lower than the experiment.
Yes.
Have you ever thought of what you would add if you wanted to correct this? And did you consider the fact that in the experimental image at the animal pole, it looks like the cell was bigger?
Yes.
If they were stiffer, then maybe they would form less locally.
Yeah, so that's one discussion we get, of course. So when I presented that, experimentalists were quite happy because it's still a good stuff. But as you said, I was quite surprised by this because you see you really have a difference of curvature there. So I tried to get some idea for why is it that and what are we missing. And I think, as you said, there is different stuff that can be. Of course, it can be the animal side. As you said, it's thicker, so maybe it's a difference of rigidity. But it's also that in our model we do the thing really simple, just by drawing an active zone there. But if you look, I think at the myosin, you see that, for example, you also have a difference between apical myosin and the basal myosin at the animal side. And if you look carefully, you also have it at the lateral side. So maybe it's just that we are not taking into account all the activity of the embryo, and we don't get the exact shape. So maybe if we add some active parts all over the rest of the embryo, we could get something that's much better. Yes.
No, and the pre-patterning was important so that it elongates to the inside and not to the outside. But
Yes.
you would deform it like passively, like you just have this deformation, and then you trigger this outer ring. You
Yes.
still deform inside.
I didn't get to the first step that you proposed.
But I understood that the pre-patterning was important to flatten
Yes.
this part so that when you have these cables
Yes.
outside activating, it elongates to the inside and not to the outside, because if you don't have the pre-patterning, you have instead an elongation to the outside.
Yes. Yes.
So is the effect of you impose this shape from the outside without having the cells in the middle, in the inner ring constricting, you will still have an imagination or not?
Okay, so what you propose is just to inhibitate the apically constricted zone and just to do the same but mechanically externally. Is that?
Yeah. Like,
Okay.
I do the cables can do that.
I expect that if you do the same shape, you should have also invagination. I don't know if it's really simple to do that experimentally. We didn't think about that. That's a good point, maybe. But yes, yeah, I expect that it's really the shape that is important. That you have some negative curvature there at the beginning, and when once you push, it's going inside. But has it to be biological, or if you are just reproducing the same shape, is it also okay? I think it should be okay, but.
Because it's interesting that in this case, the shape is making, the animal is making its own shape, but sometimes it's the outside that puts a shape, so then you don't need to.
Yeah, for sea urchin, it's really a free embryo, and it has nothing to do with the outside. So the answer is really inside of the embryo. You don't have any egg or something like that. It's really free. So it has really to be a self effect.
My question, I have two questions that are related. So the first one is on the acting cables.
Yes.
In the image you showed them, I didn't necessarily see them. Like, I could have imagined many more ways to draw them. How essential are they? Are you in the experimental that they are there? And if are there alternative ways, for example, you mentioned pressure that could help do the same process. And the second question is, how do you think that the system stops? The imagination stops. It seems that your imagination was going farther, so compared to what you had in the experiment.
Okay, so for the first question, okay, so I think I don't know. There is quite what is important there is that you have an anisotropy that is really creating you an anisotropy in the tension of the torque. And okay, so maybe you can say that the orientation you are not so confident about that, but there, if you are doing some laser ablation, you really see an anisotropy in the tension. So you will see that you have much more tension circumferentially than radially. So it means that somehow actively you are creating some anisotropic stress there. What is the real mechanism about that? I don't really know, but I think that this orientation of the cables are quite a good explanation for that. How they are doing this, I don't see, but you really see that you have some kind of more and more orientation of your cables when you are following one.
So maybe I'm not too, maybe I don't know exactly the definition of actin cable. So is it just the fact that some actin enriched cortex membrane is aligned, or is it really a supracellular thing?
I think for me it's more that it's aligned. Our collaborators speak of supracellular cables. I'm not really sure how we can say it's supracellular, but for me the important point is that it's really an orientational effect. And the second question was about
Imagination, like, there you go. but
yeah.
That's more than the cell data, right?
Okay, so there, if you go there, actually, it's just crossing the animal pole. If you don't stop it, it will never stop because they are not feeling one another, of course. Okay, so it's an important point that we didn't fix for the moment because for our collaborators, they say there is two phases in the invagination. You have a first phase that is quite quick, where the invagination is deepening quite rapidly. And after that, it seems that you have a second phase where it's much slower. And in particular, in this second phase, if I show you this, I didn't speak about that because it's not our point. But you see there in this scheme, in the second phase, the cells that are at the tip are doing some philopodia. And the philopodia are at some point reaching the opposite side, just tucking and pulling also. So there is really a different mechanism to get the tube reaching the opposite pole. And in some other experiments of other species of sea urchin, they see that if you cut those philopodia, the invagination stop at some point. It's not reaching the animal pole. So in our case, I just show you the ideal case where we are reaching the animal pole, but playing on the amplitude that you are applying, you see that at some point it's not reaching the animal pole. There is kind of a buckling instability. At some point you are not doing anything, and when you overcome the threshold for the buckling instability, your stuff is directly going to the animal pole. So what we consider is that the first phase can be that, but after some time there is some biological change on this outer ring. You see, it cannot be so far in the tube. It's not obvious that they should also still be active. So maybe at some point they just are stopping being active, and it's some other mechanism that is playing. Yes.
So basically, you consider that lacking the blood in the minus. And you ever consider that it's also possible that our size and ours can also contribute to the who then has the thinking
Okay, so there actually we don't only work with actin. We work with the myosin microscopy to have a visual explanation of what is going, but what is driving us is much more the micropipetting tension measurement. And that it's taking into account whatever there is. It's not just the actin. It's really giving you the exact tension you have at an exact point. And what we see, it's really that you have kind of three region in your embryo. You have the lateral one where you don't have so much tension, and at the animal pole you have some tension. But if we are not adding that to the model, we still recover what we get. And what is really important is that part there, the vegetal part, and we try to fit the model with this measurement. So we are not working just with the actin actually. And okay.
So in the model, you like the growth, you only see in the external figure, and you do this simulation, it looks like you still need to do contribution. But instead in the video, to make it looks more like animation vision, like the whole issue bending.
Okay. I'm not sure I get your point. So, okay, you mean there is no growth and it's just
going inside, and instead in the simulation it looks like that the outer ring is growing.
Yes, okay, so okay, I get your point. Sorry. Yes, so you mean the anisotropic region is growing. Okay, so in reality, it's not growing because you have to think that it's an axisymmetric surface. So what is important is the area, and the area here is conserved. The area of the beginning is the same that at the end, as it's anisotropic. It's not changing the area because it's going to be closer to the axis of revolution. So the area is the same, but it's clear that if you try to fit that with the data we have, it means that you have more and more cell going to be active. So that's a limitation of our model for the moment. When we speak with our collaborators for the moment, they suspect that the activity is just at the shoulder. Once the cells are inside, they are no more active. So to be exact, it's what we should simulate. It's what I think what we are going to do next. But here it was really to have some elements to show that just putting anisotropy is doing the job. But yes, if we want to be more exact, it won't be like that. And this last part, it's really not relevant with the experiment. You never have something so thin, some tube so thin at the end. So yes, at some point we should have something that is cutting the activity there. So yes. Okay. Thank you.
There's 3 10 outside and 5